Hey, it's Miss Garcia coming at you. Today we're going to be talking about Punnett squares. So Punnett squares are these little boxes that you see here. And what they are, it's a diagram that basically can be used to predict future offspring or like future possible outcomes of children between two individuals. So it can kind of predict their traits. It can give you percentages like maybe something like, oh, 75% of the offspring would be tall, 25% of the offspring has a probability of being short, things like that. So we're going to run through some examples today, starting with some examples where the parents' genotypes are given. So in this example here, the two parents' genotypes are given right here. So you'll see the way that these genotypes can be expressed. So is they're both heterozygous. Remember, heterozygous is when they have two different letters. So it doesn't matter which parents, what. So it doesn't matter if this is mom, this is dad. No matter where you put parents on the Punnett square, the outcomes are basically gonna turn out the same. Okay, so let's get to it. And remember, you're gonna get, so this capital T here, like, let me draw this here. Capital T, lowercase t, that's a genotype, right? But the letters by themselves, those are alleles. Alleles are a variation. And whenever offspring are whenever gametes are being formed, the alleles separate out. So they don't go together hand in hand because you're going to get one allele from mom, one allele from dad, and then your two alleles combined will be your trait. Okay, so let's start making sense of this and working out some problems. Okay. So in the first problem here, these are the parents' genotypes. And basically the way you fill these squares out is that the parents' genotypes are going to go on the outside of the square, on the top and the left side. And then the offspring, their genotypes, will be within the square after you work out the problems. So it doesn't matter what parent goes where. Like, for instance, I'm going to put this parent on the top here, and I'm going to put this parent over here on the left-hand side. The outcomes, again, will be the same. So I'm going to put a capital T here, lowercase t here, and then I'm going to put capital T here, lowercase t here. Excellent. And so now the way that you fill this out is you're going to pull one allele from this parent, one allele from this parent. So I'll have two capital T's inside this first square. Okay, in my next square, I'm going to have one allele from this parent again, one allele from them. So then I have one capital T, one lowercase t. Okay, this square, I will pull one allele from this parent, one allele from that parent. So then I have capital T, lowercase t. And then again, I'll pull one allele from this parent, one allele from that parent. And then I'll have two lowercase t's. Don't forget, when you're filling out these squares, also capitals always come first. Okay, so I'm all finished up here, but let's just review some vocabulary that you really need to know, especially when it comes to word problems. So the first vocabulary term we need to talk about, all the genotypes within this offspring. So genotypes are the letters that are used to represent their genes. So this genotype, the way to express that out loud, because they're both the same letter, remember that's homozygous, and since they're capital T's, this offspring would be homozygous dominant. This offspring, since the letters are different, are heterozygous, so this one's also heterozygous, and the way to express this is homozygous recessive. Okay, so now that we've reviewed, I'm done using capital T, lowercase t, excuse me, and I'm going to use homozygous, heterozygous, that sort of thing. Okay, so let's look at our possible outcomes over here on the right. Okay, so how, out of our possible outcomes, how many of the offspring would end up being tall? What's the possibility of the outcome, or what's the probability of the offspring being tall? So look over here, and if they have at least one capital T, that trait is going to show up, because remember, dominant masks recessive. So that would be 1, 2, 3 individuals who would show up as being tall. So 3. And as a percentage, that would be 75%. So 3 out of 4 is 75%, right? And if you're not a math person, think of this over here as being a dollar, right? 1, 2, 3, 4 quarters add up to a dollar. So 1, 2, 3, 4, 1, 2, 3 quarters, that's 75 cents. Okay, so looking over here, how many would end up being short? So only one of the offspring have the probability of being short. So that's 1. So that would be 25%. Okay. Okay. So now this part right here, the genotypic and phenotypic ratio. I would say that this might be the most difficult concept of all of this is coming up with the genotypic and phenotypic ratio. So at the top of your note sheet, we're going to write a little formula to kind of help get you through that. So what the genotypic ratio is it kind of wants to know out of the 3 possible genotypes, you could have a homozygous dominant genotype, a heterozygous genotype, or a homozygous recessive genotype. It wants to know how many of each of those will you have in comparison to one another. So let's go to the top of our papers up here, and we're going to write ourselves a little cheat sheet. So we're going to write genotypic ratio genotypic ratio. And so what the genotypic ratio wants to know, this will be our little formula, how many will be homozygous, of the offspring will be homozygous dominant, versus how many will be heterozygous, versus how many will be homozygous recessive. So let's go back down over here and check it out. So how many do we have at the bottom that are homozygous dominant, like that? So that we have 1 that's homozygous dominant. So write that here. How many offspring are heterozygous, like this? We have 1, 2 that are heterozygous. So we're going to write 1 to 2. And then how many do we have that are homozygous recessive, like this? We have 1 offspring that's homozygous recessive. Excellent. So our genotypic ratio is 1 to 2 to 1. Okay, so now let's figure out what the phenotypic ratio is. We're going to write another cheat sheet over here at the top. So phenotypic ratio, we're going to erase this little cheat sheet. Let's see. Our phenotypic ratio use red. So phenotypic ratio is how many of the offspring will show up with the dominant appearance versus how many will show up with the recessive appearance. So how many will show up, in this instance, tall versus how many will show up short, which is the recessive. And so the problems will usually tell you what's the dominant versus what's the recessive trait. So how many show up with the dominant trait over here, at least having 1 capital T, 1, 2, 3. So we'll write that down. We've got 3. And how many show up with the recessive appearance? 1. So our ratio is 3 to 1. Okay. Next problem. Let's head over here. We're going to run through this one pretty quick. Okay. So we have a heterozygote with a homozygous dominant parent. Doesn't matter who you put where. I'm going to put my first parent here, my second parent here, and then I'll cross it out. Okay, excellent. And then so how many will end up being tall? And so I have 1, 2, 3, 4 with capital T, so that is 4 or 100%. Okay, 0 being short. The genotypic ratio, remember we can see it up there at the top. How many do we have that are homozygous dominant? We have 1, 2. How many do we have that are heterozygous? We have 1, 2. And how many do we have that are homozygous recessive? 0. Our phenotypic ratio is how many do we have with the dominant appearance? That's all of them, right? We said all of them were tall. So that's going to be 4 to 0 show up with the recessive appearance. Okay, so those are the easy problems when the genotype is given to you. So let's go on to our word problems. This is why I said vocab is super important. So over here it says cross a homozygous axial plant with a terminal plant. Homozygous axial with a terminal plant. So up here, we go back. Up here it tells you axial is the dominant trait. So homozygous axial means that you have to have 2 capital T's because that's the 2 capital A's, excuse me, because that's the dominant trait. So that's the first parent's genotype. And then the other genotype says that it's with a terminal plant. So you see where it says terminal? The only way you could be terminal is if you had 2 lowercase a's. So that parent is homozygous recessive. Excellent. So let's go ahead and cross it on out. I'm going to put one parent here. And that's an A. Oof. And one parent here. Okay, cross it out. One allele from mom, one allele from dad, one allele from mom, one allele from dad, one allele from mom, one allele from dad. A stylus would be nice. My handwriting's gross. One allele from mom, one allele from dad. All right, we're all crossed out. So what percent would be tall? Look at that. Everybody's got a capital A. So 4 would be tall. That's 100%. And 0 would be short. That is 0%. Okay, genotypic ratio. Remember, look at the top for your cheat sheet. And you'll keep using this as you do your practice problems. So we have 0 that are homozygous dominant. We have 4 that are heterozygotes. And we have 0 that are homozygous recessive. And then we have, for our phenotypic ratio, basically how many show up with the dominant trait, how many show up with axial flowers, 4, how many show up with terminal flowers, 0. Okay, on to the next. Next problem says we have a cross between 2 plants that are heterozygous for axial flowers. Remember, heterozygous means 2 different letters. So up here, we'll do 2 different, they're heterozygous, so they have a capital A and a lowercase a. And doesn't matter what parent you put where, remember? Cross it on out. Alrighty. So now, let's check out our possible outcomes. We have, how many that end up being tall? We have 3 that all have a capital A, the dominant trait. So that's what percent? 3 out of 4 is 75%. And then what percent do we have being short? There's just this one offspring right here. So that's 1 out of 4. That's 25%. Dope. So genotypic ratio. How many do we have that's showing up homozygous dominant? 1. How many do we have heterozygotes? 2. Homozygous recessive? 1. So that's 1 to 2 to 1. Okay, lastly, our phenotypic ratio is how many do we show up, or how many show up with the dominant appearance? That would be these 3 with capital A's, 3 to 1. 1 shows up with the recessive appearance. 3 to 1. Okay. All right, now we're moving on to the big leagues. So, in these last couple of problems, basically, the parents' genotypes are not given to you. You're going to have to figure out what the parents' genotypes are. So this is talking about pea color, or pea pod color. So green is the dominant color, yellow is the recessive. So this problem says 3/4 of the plants produced by a cross between 2 unknown pea plants have green pea pods, and 1/4 have yellow pods. What are the genotypes? So these 3/4 have green pods. Check it out. Green's dominant. There's 2 ways that could show up. You could be homozygous dominant, so 2 capital G's, or you could be heterozygous. one capital G and one lowercase g together, still going to have green pods. So the only thing we know for sure about any of these offspring is this 1/4 right here has yellow pods. So in order to be yellow, the only genotype you can have is 2 little g's. So we are going to start the cross by go ahead and putting that. We know that. So those 2 little g's are going to go right here in this corner. Actually, that's a good rule of thumb when you're filling out a Punnett square backwards. If you're trying to fill out the genotypes, anything recessive is always going to go on this bottom right corner, because remember how capital letters go first. That's the way that it's going to land, where lowercase letters are always at the end. So if you have any recessive traits, they'll go in the bottom right corner. If you have any more than one, the next recessive trait was going to come over right here at the bottom left corner. And that would be it, unless the whole cross is recessive. Okay, so now let's go on. We know that, and remember, we know that 3 of the 4 have green pods, so we at least know they have 1 capital G, so we can fill that out. So 1 capital G, there's another capital G here, so there's 3 offspring. Okay, so now we work backwards to try to figure out the parents with what we know. So I know this little g had to come from here, and I know this little g had to come from here. So let's fill out those parents. Little g, little g. So I know this also has to be little g. And I know that this also has to be a little g, meaning that capital G right here had to come from up here. So we're going to put a capital G here. And then this capital G had to have come from this parent. So we are going to put it here. And then we can finally finish filling out the rest. So the genotypes of the parents were, one was a heterozygous and one was, well, they were actually both were heterozygous. There it is. Okay, next one. Okay, so what would what cross would result in 1/2 the offspring having green pods and 1/2 having yellow pods? So we know for sure what the genotypes are if they're yellow, right? So let's see. These guys right here, yellow, has to be 2 little g's, right? So that's where we're going to start. I'm going to write my 2 little g's. And then it says 1/2 has to have green pods, but we don't know if they're dominant or heterozygous. So all we can put is the capital G and work backwards from there. Okay. So I know that this little g had to come from here, and these little g's had to come from this side. Okay, so that can come down here, meaning the capital G had to come from over here. And this little g had to come from up here. So the parents' genotypes are, one was homozygous recessive and one was heterozygous. Okay, last problem. We kind of didn't give you all the 411 here, all the info. So I'm going to give it to you now. Let's say that there were 100 flowers. Kind of left that part out when we were making this. 100 flowers. 73 of the flowers. Oh, this one's right. 73 purple flowers and 23 white flowers. Okay, so that adds up to 100%, or 100. So we can think of that as being 100%, so that should help us out with the cross. So 27 white. So we know for sure their genotype, right? I know that one box is 25, but not 27. Well, guys, in nature, things aren't going to be perfect. So if this was like a real life example, you know, we're going to be growing corn in our class. We started doing that the other day. It's not going to be 25 exactly. It's, you know, should be close to 25. So I'm going to know that this number is close enough to 25 to be my 1/4 that goes here. So white flowers, white flowers are going to go here as the 25%. And my purple flowers are going to go everywhere else. Okay, so then I work backwards. Little p's here, little p's here, and then my little p here. Big P had to come from here. Little P here. Big P had to come from here. All right, we're done, though. So they were both heterozygous. Oh my gosh, that's a Q, not a P. Okay, so, all right, check out. I have one more point to make. If you ever get to pick your letters, so if you're coming up with your own genetics problem and you get to choose, you see how confusing it is when you choose a letter like P where they look the same from capital to lowercase forms. Choose a letter that's really distinctly different. Like an A is very different from its lowercase to capital form. All right, friends, thanks for joining me. If I went too fast, sorry about you. I was trying to get you done in a timely manner. Just press rewind. Thanks for watching. Good luck.